This podcast is sponsored by Echelon. Echelon is the affordable way to get the workout equipment, the workout community, and an instructor's motivation right in the comfort of your own home. With Echelon, you can work at any time, day or night, and crush your fitness goals. And right now, for a limited time, podcast listeners get up to $800 off MSRP. To get this exclusive podcast discount, text GENIUS to 818181 to get up to $800 off MSRP. Once again, just text GENIUS to 818181. Quick disclaimer, message and data rates may apply. See terms for details.
Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field. Sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. I have a returning guest, Henry Heng. He's the part of the Center for Molecular Medicine and Genomics at Wayne State University. He's been my guest before, very interesting guy. And now we're joined by Rafe Furst. He's a founder, investor. He was part of the World Series of Poker Champions. But we're going to be talking about biological type stuff. So I want to welcome both of you. Thank you for coming.
Thank you for having us.
Yeah, if you would, Rafe, since you're brand new, and then we'll ask Henry. Rafe, tell me a little bit about your background and how did you end up working with Henry?
Yeah, that's a good part of the story. So I was not at all interested in biological sciences or medicine or even cancer. I was a computer scientist by training and was in Silicon Valley during the 90s internet boom and became an entrepreneur and an investor. And I was a poker player, as you mentioned, and I, along with another Silicon Valley poker player, Phil Gordon, got interested in cancer, mostly from a prevention and early detection standpoint. We both became board members of the Prevent Cancer Foundation. And it was on my tenure there on the board that I was introduced to Henry and a whole host of cancer researchers who were focused on the evolutionary mechanisms of cancer. And I had a lot of catching up to do, and I was fascinated. And I really gravitated towards Henry because he had some really, I would say, heterodox views at the time, in contrast to the orthodox views of what cancer is and how it progresses and what we might do about it in terms of treatment. And I knew about the urgent need because at the Prevent Cancer Foundation, we were very aware of the billions and billions of dollars that have been spent for decades trying to eradicate cancer and cure cancer, and to no avail. The age-adjusted mortality of cancer has not moved significantly at all in the 70 years that we've been keeping these statistics. And we're not really told this. This is not what the story is.
Well, can you confirm a stat that I've heard that is probably the most troubling of all, I think, that 50% of U.S. men and 33% of U.S. women will get cancer in their lifetimes?
I don't know. I haven't kept up with the stats. Henry, what do you say?
Yeah, correct. Yeah, go ahead.
Mm-hmm. Yeah, when I hear that just like, I've had cancer before too, thyroid cancer, thank God it wasn't too bad. But, if that stat's anywhere close to accurate, that's completely devastating. That's really worrisome. So I just wanted to point that out.
Absolutely.
Hopefully that's accurate. So,
no, I was just going to point out that, even from my perspective, yes, that is devastating, but also it seems likely from where I stand, and Henry, you can weigh in separately if you agree or not, but by and large, there is a decent chance that the treatments that we're using to try to kill cancer actually are doing more harm than good overall. So, yeah.
Well, we'll get into the content more in a minute, but Henry, I just wanted you to get the chance to give your little bit more extended bio, and then I want to ask you guys about your current research. So Henry, go ahead, please.
Sure. So my research initially is study the cancer progression, and especially we try to find the molecular mechanism to understand the cancer. But during our research, we actually realized it's not simply molecular mechanism because there are so many of them, and the highly dynamic make it's extremely challenging to predict the disease progression based on a particular molecular mechanism, which is the main focus in the past 50 years. So we actually realized that we need to use the evolutionary idea to study it. But during such a process, we actually realized the conventional conceptual aspect of the evolutionary theory, genetic theory, actually had some potentially major problem. So that's a story, started our research. So on one hand, we study cancer. On the other hand, we use cancer as a model to try to understand whether or not the concept of evolutionary and the genetic research actually is, as we thought, as correct, but it turns out to have some of major problem. So that's the reason we actually team up with different people to try to promote such idea. And Rafe and other people, they're outside of the field, it seems like they're more open-minded. So that's a link between different group people to talking about that, because they're not closely associated with the field. Maybe people within the field is too near to judge. So that's one of the rationale we actually love to team up with different people with different background and thinking style. Yeah.
Okay, so currently, what is your research involving? And then there's a paper that recently came out, with you two that I want to go into. But tell me what's the latest and greatest in terms of your research. What questions are you both trying to answer?
So what we just step back, we actually, because of study the cancer, use cancer as a model to study evolution, we realized that some of the basic assumption in the traditional evolutionary sense seems like it's not working, right? So for example, one of the key idea is evolution is driven by accumulation of the small change. You accumulate after many generations, then you become some totally different system. So that is one of the key thinking from Darwin's time. But use cancer as a model, we found maybe this is not the case, because the small change which mediated by gene mutation is quite different from the big change, like mediated by the chromosomal reorganization. So those discoveries really challenge the current cancer research. If that's the case, then we should use a totally different way to study the tumor progression, rather than stepwise one gene at a time accumulation become cancer, but they can drastically by reorganize the genome from the cancer. So that actually requires different approach as well as the experiment platform. For example, if we want to...
Quick question here. So, people see pictures of DNA as the double helix, and then, fewer people will see chromosomes. But to go from the picture of a double helix just hanging out there to what a chromosome looks like, what kind of structure has been added to create a... Yeah, and then in between chromosomes, how do they link up together?
So that actually is one of the crucial questions, because in the past many decades, since the discovery of the DNA, people always thinking the genetic information is all about how the information from DNA go to RNA, go to protein, right? So the gene-centric idea is if we know the gene, individual gene, then we actually can know the function of the system. But now we actually realize that, especially use our genome architecture theory, we realize there are two types of basic information. One is parts information, right? Like for example, particular gene responsible for the protein or for the certain regulation. But above that, there is another higher level of information, which we call the system information, which is coding beyond the gene. It is something to coding how the gene interaction can work. So that's we actually figured out is all about how the gene, the physical relationship with each other on the chromosome. That is a new type of information we call the system information. It turns out that system information is the basis for the cancer evolution, including for the different diseases condition. For example, the neurodegeneration diseases, we think is also an information problem diseases, rather than individual gene, but how the system can handle, can inheritance, and create new information to driving the new system. So that's all we actually try to discuss about.
Yeah, just to break it down, I guess more simply, you talked about chromosome rearrangement as part of cancer, but for the layperson, what does that look like? Like the chromosomes look like little X's and L's, I guess, that are furry, and the DNA is all wrapped up there, et cetera, around histones and all that. But are two chromosomes that are adjacent, are they literally connected to each other? And when they rearrange, what does that look like? Again, just in a real layperson level, what do you observe under a microscope that looks different, before and after?
So, for example, we use one of technology to study the visualization of the whole process. So we can paint each chromosome with one of a particular color, right? So, for example, chromosome 1 is red, chromosome 2 is blue. So if they have chromosomal reorganization between chromosome 1 and 2, then you will see the red and the blue color become chimeric on the chromosome. So that you immediately can visualize, they have part from 1, the red color, and the jumping on the chromosome 2 is the blue color. So the 24 chromosome have all different colors. So then you can see when they have a massive genome reorganization, then immediately you can see all the different chimeric color on each of the chromosome. So okay, the chromosome actually had the reorganization.
If a chromosome looks like an X, I'm just making this up, you can see that it has certain color patterning, I guess, on a microscope. And when it rearranges, does the whole X break apart? Does the X transfer monolithically and swap positions with other chromosomes? Or do pieces of it change shape? Like what do you literally observe that's different physically about them?
So basically just the different fragments, the reorganized fragments, right? So if I have the metaphor, we can use the building the house, you can use the same brick, but then you have different arrangement of the same brick, therefore you have different shape of the architecture. So Rafe, do you have some of the more common metaphor you can cut in?
Yeah, I mean, I think to tie it together with what Richard you're asking about on these visualizations of the chromosomes, you can imagine that one of the legs of the red X gets transposed onto the blue chromosome, and maybe vice versa, or maybe it gets reorganized with parts of other chromosomes. And there's a number of standard categories of chromosomal reorganization, transposition, copy number variation, aneuploidy, they call it, where a chromosome is either has more than the normal 2 copies within the nucleus, sometimes many more. And so I guess that the coding regions of those chromosomes, their effects are amplified when you see that. So, Henry, are there other examples of chromosomal shuffling, chromosomal chaos that you could highlight?
Yeah. So for example, if we use the real species, right? Some of animal, so like the chimpanzee and the human. Chimpanzee have the 48 chromosome, human have 46 chromosome. So 2 of the chromosome of chimpanzee, they just fusion together and become new chromosome. So if we use the sky, you actually will find the one new formed chromosome that have 2 colors. So from 2 individual chromosomes, they form one of the chromosome. Half is red, half is blue. So that's a visualization on the microscope when you're doing experiment.
And sky, the terminology, sky stands for spectral karyotyping. So that's the visualization technology that Henry was referring to.
Yeah, but for the cancer chromosomes, then you just say it's totally different color. For example, one chromosome you have 12, 14 colors on one chromosome. So different region is different color. And we know the different color means they come from other pieces from other chromosomes. So they're just reshafting this relationship. So the gene is still same, but you located on the totally different chromosome and totally surrounded by.
So if someone has, let's say, liver cancer, and you biopsy the liver, and you pick up some cancer cells, but also some healthy cells, same cell type, and you do this spectral analysis, and you compare them, what do you see? Can you tell which rearrangements occurred and kind of go backwards from A to B, from healthy to unhealthy, and see the path that the cell took?
Correct. For most of the cancer, they all involve this process. So after you sequence, you always can see, if you read the sequence paper, they all have the pie chart. They have different line between different chromosomes. Every time you see the line cross the chromosome, that's one chromosome translocation. So you can see massive of them. Also you have the copy number, polyploidy, aneuploidy. In short, just a genomic chaotic change. So that's what we use the term to summarize all the different type of them.
Yeah.
It's really messed up.
Yeah, and just to highlight that, chaotic in this case doesn't mean completely random, but it definitely does mean it's non-deterministic, meaning the pattern of reshuffling that you'll see varies from cancer to cancer, from patient to patient, from tumor to tumor. And then, correct me if I'm wrong, Henry, but when you run experiments with individual cancerous cells, like in a culture, in a petri dish, and you sort of rerun the cancer evolutionary process, you won't see the same pattern twice. But what you will see is this progression of increasing diversity and chaos within the structure of the genome that is characteristic of all cancers.
Yes.
Okay. What else can you tell from these rearrangements? So they don't appear to be completely deterministic. Do they, if you looked at, again, I'm just making this up, healthy liver versus cancerous liver cells, and you look at 100 different pairings in 100 different people, will you see generally the same rearrangements, or is it like wildly different in each person and in each cell? Like, how is this totally chaotic, or what does it look like?
Sure. So initially, actually, in some of the blood type of cancer, right? So they have, with some exceptions, some like CML, so some of kinds of leukemia. So the 95% of the patients, they have similar chromosome translocation. For example, they called the Philadelphia chromosome, right? That's the chromosome 9 and the 22 translocation. So for that particular diseases, you have a high frequency of recurrent translocation. But for most of the solid tumor, every individual patient, the most different combination of the chromosomal change. So for a long time, we have one exception, and in all the field, people try to find the same success to identify, the same signature of translocation. But it appears for majority of the solid tumor, it's very difficult to find that. Now we know the reason, because as soon as the chromosome translocation become new system, and then the next step.
Henry, just before you go into that, because I think it's really important to highlight that you highlighted the blood cancers, in particular CLL, and these are cancers that respond really well to the treatments that we know about relative to solid cancers. Is that true?
Yes, because in that case they have a particular molecular target, right? So, but just for that diseases, it responds very well. But after your treatment for a while, as soon as an additional chromosome start to change, all this drug is no longer useful. So therefore, when the genome become chaotic, the way we are try to stop them become powerless. So that's one of the important information.
Yeah. So just at the more process level and meta level, some of the reason why we haven't made more progress overall is that we've become fixated on the relatively deterministic ways to treat certain cancers, specifically the blood cancers like CLL. But those are not representative of the majority of cancers, which don't respond well at all to the kinds of treatments, to targeted treatments. And Henry will go into, why that is. But, I think your question, Richard, was about what patterns are we seeing? We're not seeing specific molecular targetable patterns. What the pattern we're seeing is a meta pattern, the pattern of chaos or massive heterogeneity, massive diversity within the genome itself. And that's what seems to be characteristic of cancer. And that's what Henry has really exposed, is this notion of genome chaos itself being a driver of evolution alongside Darwinian evolution. So I'll just pause there. Go ahead.
Just a couple more questions about the mechanics of this. If you look at a population of healthy cells, how much variation will there be in the patterning of their chromosomes to establish a baseline? Let's say, again, in a particular tissue where blood's probably the most accessible. Have you guys done that to see, again, what the baseline looks like before this variation?
Sure. We actually did such a study. So is different individuals that have very slightly different, but the rate is extremely low. Usually, if you check the 100 cell, they have one or two have some change, but this randomly change, right? But for the cancer patient, especially if after the drug treatment, the increase dramatically for the random change rate. So you can easily reach the 20, 30, 40% of the cell. They all have different craziness. But those kinds of change is difficult to be identified by the sequence technology, because the sequence them, you have to get the average of the cell population. So you only can be identify this heterogeneity based on single cell technology, because when we study chromosome, we actually study each of the chromosome as the cell as a unit. So we can.
Yeah, but even if you do single cell, if there's just a rearrangement and not necessarily a reduction or an addition of certain chromosomal segments, it probably wouldn't even appear on sequencing either, because it breaks stuff up into fragments, so far as I know, long or short fragments, but you may or may not catch nearly all the variation or maybe any of it.
So that's why we try not to focus on one cell. So we actually say how stable the population is. So now we actually realize that if we have certain degree of population, they all have the cell had a problem, then you had a bigger problem in terms of evolutionary potential. So that's actually become very important. For example, in Europe, people actually did the experiment. They studied the chromosome of not just the patient, but the patient's relatives, like brothers, sisters. They don't have cancer. But however, if you compare how genome stable, they're actually already much more unstable than the normal population group. So therefore, now we actually realize that when the system is unstable, you have to have all the chromosomal change, but not most of them will lead to the cancer, but just a very tiny proportion of them will become the cancer. So that is extremely difficult for us right now to use molecular signature to predict. But that's what we have been working hard to try to push the idea. If we can monitor the evolutionary process themselves, then we actually can tell people which situation is more likely will have the success of the cancer. So that's one of the idea to really push. And you asked a very important question. So we actually categorize different chromosomal change, cell change. We will say how complicated this change is and what's the overall frequency of the population. In fact, we already found if you just check different type of cancer people, different the prostate cancer, breast cancer, they all involve the overall genome instability. So that could be one of the key links to understand why someone have cancer, someone don't, is because when the system under stress become unstable, the evolutionary game started, but only a small proportion will finish the game because it's not easy to become cancer cell. Every cell wants to kill you. So you have to keep changing, and after the perfect storm, you become cancer. Yeah, so that's
based on what you guys see, are there any epigenetically created like nucleation sites that would cause this rearrangement? Or again, are there any naturally occurring rearrangements or other features of the chromosomes physically that would predispose them to rearranging in the right or wrong way? I've been working too hard and not working out enough. I wanted to get in shape, but I don't have time to get to the gym. Echelon brings the gym home to me. So right now, for a limited time, podcast listeners get up to $800 off MSRP. To get this exclusive podcast discount, text GENIUS, GENIUS, to 818181 to get up to $800 off MSRP. Once again, text GENIUS to 818181, and message and data rates may apply. See terms for details.
That is, again, it's a very good question. So we know, first of all, every individual have a certain degree of random chromosome change, right? So for a long time, we was puzzling. We thought, people always thinking, this change is a mistake. You have error of the replication. But now we understand, actually, this random change have its biological meaning, is essential for the cellular adaptation, right? So, for example, after you have the virus infection, your genome start to change, because only that way they actually can handle the virus infection, right? So if you have the wound, if you damage the tissue, the chromosome start to change, because in that way they can very effectively repair the damage. So when the liver start to, the metabolic process, the chromosome have to change, because if you do not change, you cannot handle the stress, so you change. So therefore, the change of the chromosome, in fact, is a consequence of the cellular adaptation. Why is that? Is it because, we have the germinized and the somatic cell separation, and every species have the same karyotype, right? But under the different situation, if the cell try to survive under stress, they have to change. So that's the key to be survived. But after change, they have to pay the price. Therefore, the cellular adaptation, the price to pay is change. And some change is diseases. So that's a very simple story right now. It's almost like the two faces of one coin.
Could you consider this to be another type of change? So there's genetic change, or at least epigenetic change and various types of it. Could this be, karyotype rearrangement? Could that become a new type of change that, happens in many different people and creatures, and also for many different reasons, maybe not just cancer?
Yes. Actually, you mentioned all the change. We actually right now realize is all about. For example, initially, epigenetic change is most sensitive, right? So you always have that. And the second level is gene change. And the higher level is chromosomal change. Is chromosomal change for that particular cell become point of no return. So they all depends on how higher the stress is, right? So that is actually, very true. So if we do not have this capability of changing the genome, then in many situations, we cannot tolerate. So the evolution actually favors the capability of evolution, despite the results is harmful, right? But that actually is quite important, because all the humans continually move forward. But if we do not, the human right now have, the occupy the ecosystem is so diverse, right? So almost the temperature, the geography, everything. So in that case, we actually have more stressful situation. So that's why, we get more of the cancer cases.
Maybe to summarize that in a slightly more abstract way, Henry is saying, correct me if I'm wrong, Henry, that normal course of life as a human, as a multicellular organism, the change at the chromosomal level, these changes at the chromosomal level help us. They're adaptive. They help us adapt to different environments, and they also help us adapt to stresses like disease from infection, for instance, maybe even emotional stresses. But all of these adaptations at the chromosomal level are due to stresses on the system, on the organism, on the human being. And it's great that we have this mechanism in us that allows us to be adaptive to these stresses. However, at a certain point, the stress can become overwhelming to the genomic system and initiate this very chaotic process that Henry can talk about. And once that genome chaos happens or begins, it starts driving somatic evolution of the cells in your body, the population of cells. And this is what we notice is clinically identifiable as cancer. So I'll just pause there. Henry, did I misstate anything, or do you want to clarify that?
Yeah, basically we can think that. So therefore, we think the cancer, is kind of a byproduct of the evolutionary process.
Is this, do you believe that cancer, instead of being random mutation driven, is it a forced adaptation due to stress that turns into a maladaptation?
This is a more complicated question because so we actually consider cancer when the karyotype changed, the system information changed. So they become different system. So the cancer almost like a new system emerged within our human body or animal body, for example. So they actually become totally different system now because they have different chromosome composition, they become new system. So in that case, we even do not agree that cancer cell is simply cheating the same type of cell. They actually is totally different type of cell. So
just to maybe clarify or put a fine point on it, what Henry, sometimes you like to say that the cancer cells, once they, are showing cancerous properties, actually have become a new species of cell. They're no longer the original human species or cells, the species of cell that is in the human body. They're a totally different system or species. Is that true?
Yes. So if we consider, how much, because this one people may think, some people think is controversial, but if you're thinking what the system is, right, what is determining the different species is the genetic, the system, how the system information are different. So therefore is the fundamentally different. That's why we could refer to them as a different cellular species. So that when the new system formed, then we cannot use a human-centric view to judge them. I mean, for the cancer, they are actually beautiful. They are doing a good job to, form the new system, become dominant, right? So, but from human-centric, also this is evil, all of this. But if we from new system emerge, almost like a human initially, we actually emerge, we all come from other type of genome, right? So that's the same fair game, and it's still going on, right? So it's difficult to really judge that part.
Well, it also sounds like the definition of speciation is too narrow, because if it's reproductive isolation, but a cell reproduces, quote unquote, by mitosis, there is no reproductive isolation. The progeny are all come from that cell. So there has to be a different definition of what is a species and what is speciation. It seems to be like woefully incomplete.
Yeah. And I think that maybe the point here, is not to say, that we should really respect and love what cancer is doing because it's surviving really well, but rather our mindset, our understanding of what cancer is needs to change, because the metaphor, a war, for instance, has dominated the thinking. We have to kill every last cancer cell. But if a cancer cell is not what we think it is, and in particular, by killing individual cancer cells, we actually induce population-level resiliency in the form of adaptation, we're actually doing more harm than good. This is what I was alluding to earlier. We induce drug resistance, and we make the cancerous population stronger. So that doesn't suggest what we should do about it or how we should treat it, but it does suggest that we should rethink how we're treating it, because we're actually not in line with the reality of what's going on in the body once we introduce cytotoxic treatments.
So back to Richard's viewpoint, I think that's a very interesting one, because in the classical evolutionary sense, especially neo-Darwinian, right? So the neo-Darwinian always thinking all the evolution is about the gene frequency change within the population. But if you accumulate more and more, you become a new species. But now from the cancer study, we realized we need to redefine this. So now we have a simple definition. If the chromosomal system change, then that's a new species. And then if just gene frequency change, the clonal expansion, and that is involve some of new feature of the same species. So now we actually realize that the macroevolution is all about speciation, and the microevolution is all about the population size, bigger or small, with adaptation. So that is one of the example why we're thinking we need the drastically change the key assumption of the Darwinian evolution, because they're always thinking everything small change, accumulation, different gene, finally become new system. But now we realize when this cancer cell dramatically changes genome, they already formed new species. But whether or not some of this will become dominant, it depends on some of the cancer gene to help them, and epigenetic mechanism to help them to get more copy. So that phase is the Darwinian evolutionary phase, right? Just a small proportion of the whole story. Yeah.
Well, it sounds like you don't understand it fully yet, but in the future, I could see that a biopsy and histology would be coupled with your analysis, this spectral analysis as well. And if you understand, what different spectral changes are associated with in terms of restructuring, that would also help to identify what's going on in someone's tumor, for instance, or tissue.
Correct, but there is from the expectation we also need to change is because the new science, right? So before the Human Genome Project, the traditional genomic study, we always hope the individual gene have highly deterministic power to predict. But now we know this is just an illusion because most of the gene we sequenced, right? They can involve all the different traits. It's not like the traditional people thinking you have A gene, you have A traits, B gene, you have B trait. It's not the case. So that's why after the cancer genome sequence project, we have all the gene mutation we cannot predict clinically very well. And in fact, the chromosomal-based prediction is much better than gene mutation-based mutation. Right? That's very surprised, but that's the results come from cancer genome sequence project. So therefore, in the future, because we need to embrace the uncertainty and the complexity. So therefore, I doubt we ever be able to predict precisely based on the pattern of the chromosomal change. Those only apply for some exception like CML or some blood cancer type. We can predict. But for overview, the science in the future, we have to accept it, the limitation of the predictability, right, for prediction. Because the new science tell us, like the chaos theory tell us, the initial condition, whenever is continually changing, you just cannot predict. So that's maybe the human fate. We have to change our anticipation about the science. And after that, we actually more comfortable to accept, the certain degree of uncertainty.
Just to piggyback on that, because I know this is something that you've highlighted in other papers and interviews, Henry, that because evolution seems to be not just a Darwinian stepwise small accumulation of changes process, natural selection, not just the Darwinian natural selection, but also this genome chaos phase. Henry has described it as a punctuated evolutionary model, which is analogous to the punctuated equilibrium model of organismal evolution that Gould and Eldridge proposed back in the 60s and 70s. Is that right?
Yes. Yeah. 70s.
Because cancer progression is this two-phased process, there are things that might work in one phase that might do damage in the other phase, and maybe vice versa. So, Henry, do you want to just expand on that and make it a little bit clearer?
Sure. So the information management is a new topic we actually also focus on, because for a long time in the field, people always thinking how to figure out the information flow from DNA, from gene to protein to phenotype. But now the genome chaos process themselves is an effective system to create new system information, to create new species. So all of this dynamic together make the treatment become extremely challenging. For example, it's very good to kill the cancer cell, right? But if it was too much power, you actually create new species that no longer can be killed, right? So we actually create the monster by our treatment, right? Another thing is for the immune system, right? Immune system is very good. They actually kill cancer cell. But somehow this cancer cell, they play the trick. They actually recruiting immune cell to help cancer cell to fight with the normal cell. right? So that actually is quite, the striking. They have all the tricks, and because maybe it's just evolutionary with the help of the evolution process, so they can change to the short-term benefit to long-term harmful. So now we actually can reverse the game. We can lose the fight of the short term and just focus on the long-term system stability. Even though the cancer is there, we actually can tolerate, let them be there. As long as they're not crazily growing, we actually use a moderate treatment to constrain the system and therefore maybe have a better outcome. So those questions we need a study. We need, more people discuss this issue.
One more thing, and it's going back into the technical side of things. This may be just helpful to you. Just a reminder, I interviewed a lady named Florencia McAllister. They were looking at the pancreatic tumors, and they discovered that each one had a localized different microbiome. So the tumors had their own microbiome versus healthy cells in the pancreas. So let's say there is no real patterning to this, genome chaos that you guys have discovered. Perhaps you could look at the localized microbiome around a tumor and infer from the changes in the microbiome versus healthy tissue what new metabolites are being produced by the cell, or what different metabolites that cell in particular needs because it has attracted different microbes around it. So just an idea.
Sure. So we last year wrote a paper talking about the evolutionary mechanism of cancer, right? So you almost can link it with unlimited, like the microenvironment, the virus domination, even nutrition, even family care, all of this. They all contribute to the complex system. But the problem is when they have so many, and you cannot identify most dominant one. So when they're doing experiment, if you only focus on microbiota, you can get a very likely story out. But if you study human, then they eat different food, different places. You use one particular way to target a specific microbiota is no longer work. That's a very, it's a very, puzzling, but it's a true fact. So for example, for all this cancer gene in animal model, they all prove it's working. If you stop them, everything is okay. For example, for a long time ago, when people use stop the blood supply, so the tumor quickly dead in the mouse, 100% beautiful story. But when apply for the patient, the large proportion of patients, when you use the same way to stop the supply of nutrition, the tumor more aggressively adopt some other way. So the people become very, very harmed by such an approach. So now we know during experimental idea, if we create a linear model, we always can prove many, many scientific power about the treatment. But when you put additional variable, it's no longer working. So that is a challenge for the complex science to the traditional reductionism challenge, because we always can figure out something and working very well, but not working in the real world, only working well in the experimental condition. So that's actually is one of the very important issue we need to discuss in the future.
Yeah, or only working well on mice or, on organoids or whatever it may be. Yeah, it makes sense. Yes. It's very complicated.
Yes, so that's exactly the case. I talked to some people that study the microbiota. I said, what's the challenge? The challenge is every time we use a human, it no longer work. But the mice, everything is beautiful.
There's an expression, I don't know who did it, that we can cure any cancer as long as it's in a mouse.
That's true.
Yeah.
I just want to clear up a point, because a couple of times, Richard, you've referred to our work. I want to just be really clear. The vast majority of the stuff we're talking about has been Henry's work over the past 40 years. I'm largely a fan and a curious layman like yourself, and I ask questions. And the reason I'm here is really because I've been just so astounded and impressed by the conceptual as well as laboratory results that Henry has come up with over his career, that I feel like it's an important message that's not getting out there. And people are dying as a result, and we're wasting many, many billions of dollars a year on approaches that Henry and others have proven over and again are not working. and will not work because they're just the wrong model. It's the wrong theory of what cancer is, and frankly, the wrong theory, or I say an incomplete theory of what evolution is.
New ideas are attacked by the current scientific immune system, just like cancer is. So
Absolutely.
I understand.
Absolutely.
Well, very good. Both of you, thanks so much for coming. I think it's been a great call. For listeners, where can they find the new paper that came out, and where can they find out more about both of you?
Yeah, Henry, why don't you suggest where?
Yeah, a few years ago, I wrote the Genome Chaos, the book, and hopefully we are going to have a new edition this year, if everything's smooth. So also they can simply check the Genome Chaos, this term, and the information creation. So they can get the information from our recent works. Yeah.
Okay, and the last item, I have a book coming out about cancer. Should be about a week or two on Amazon, and Henry is one of the co-authors in it. So for anyone listening, if you also want to see some of Henry's thoughts and answers to some of my questions that we did a while back, that'll be on Amazon. It's called Finding Genius, Understanding Cancer. Again, not to take away from Henry's work, because he's the one that's been the workhorse for 40 years, not me. But I just wanted to mention that. So again, in passing, thank you both for coming, and it's been a great call.
Thank you.
Thank you, Richard.
Yeah.
Thank you for listening. I hope you enjoyed the podcast, which has been sponsored by Echelon. When you're trying to reach your fitness goals, it can really help to have world-class instructors like Nicole Griffin and Michael Brown choreographing classes with music from your favorite artists like Pitbull. And you get a community of hundreds of thousands of people who can give you that extra push. Echelon gives you that. Echelon's certified fitness instructors are supportive, engaging, and fun. They really know how to get you moving. And right now, for a limited time, podcast listeners can get up to $800 off MSRP. To get this exclusive podcast discount, text GENIUS to 818181 to get $800 off MSRP. Once again, text GENIUS to 818181. Message and data rates may apply. Please see terms for details.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.